Good evening, friends. Never do anything you don't want to explain to a paramedic was the sign we just had up there a moment ago. Couldn't resist that. Thank you, Tamara, for sending that my way. And we're going to be involved in Technical Thursday. So I hope you got your seatbelts fastened, your pencils sharpened, your ink well full, and we'll get into it. We are going to talk about some of the research into remedies, into some of the research into vaccines for COVID-19. We won't really go into the silver tonight. I'm going to hold that possibly, and I stress possibly. We'll talk about colloidal silver and structured silver on Sunday. I'm still digging deeper into that, and I really wanted to focus on let's call it the laboratory research that's going on regarding a vaccine and a therapeutic remedy for those that have already contracted COVID-19, that two-pronged approach we talked about yesterday. The reason for that is I sent to you a couple of hours ago before the live stream two fairly significant announcements, one coming out of Canada and one coming out of Israel, both of which the laboratories there indicating that within a matter of months they may have an actual vaccine and a therapeutic treatment for those that have contracted COVID. So I really wanted to focus on that because that news is really fast breaking. It's what a lot of the media, I'm sure today, are going to latch on to. It just broke just about 2 hours before airtime this evening. So we'll go through that, and I've shared that with the patrons at the time that the articles were published. And for those of you that are not patrons, because this will be made public probably tomorrow, this broadcast, there's a lot of information that never hits the live streams that I share with the patrons. And that's typically deeper discussions, deeper details, white papers, research papers, what I consider well-vetted sources of information from news sites that I pass on to the patrons. And so if you want a more well-rounded discussion, articles, references to the things that we present in 2 hours in these live streams, then I would encourage you to join us on Patreon, okay? Alrighty, so lots of people in chat this evening. You guys are really in good spirits tonight, which is great. You usually are. But I like to see the joking around going on in the chat room before we go live. And for those that are just watching one of these live streams for the first time, just kind of a programming note. Typically, 30 minutes before airtime, I would play several pieces of music just to kind of get things warmed up. This evening, I shortened that down to 15 minutes before airtime. But it's easy enough. Fast forward, and I put a note in the caption area of the live stream that, just fast forward past the music and you'll catch up to us, okay? So, anyway, without further ado, we are going to be talking about RDRP, which is RNA-dependent RNA polymerase, of all things. We're going to talk about a treatment regimen, if you prefer, called remdesivir, and we introduced this briefly in last night's live stream. That is spelled REMDESIVIR, remdesivir. That's the primary thing that I'm going to focus on this evening. It's a way to introduce DNA and RNA because, as you know, COVID-19 is an RNA-based virus, and its genome sequence we've presented several times in past live streams. And therefore, it's important to understand DNA, RNA, focus on RNA, how it replicates DNA, what are the components, but without getting too verbose about it, I'm going to focus on just RNA and how it goes through this process of replicating DNA, again, at a very introductory level, at a level that is specific to the vaccine, specific to the remedy, the therapeutics, because it appears that one of the best opportunities for a vaccine is with this remdesivir. Remdesivir, it binds to ATP, which is adenosine triphosphate. And that's really where we're going to focus when we talk about nucleotides and amino acids, when we talk about where's the action really taking place with the building blocks of DNA. And it's with ATP. And that, again, we're just kind of giving you the heads up where we're going to focus tonight. But there's this interchange. There's a dynamic between this RNA-dependent and RNA polymerase, which again is abbreviated as RDRP. Now this may seem like much ado about nothing. Like, why am I spending all this time talking about RNA and DNA? When really, for the most part, most of us are not involved in the research, and we have no influence over who's going to come up with a vaccine. So it's always back to that question I always pose, what's in it for me? You know, that's kind of our subconscious question to a lot of things in life. What's in it for me? What does this have to do with me? Well, in most of the topics that we present here, my rationale for going deeper, getting into the deeper aspects of a particular topic from a scientific approach, is that many times in the mainstream media, a lot of unfamiliar terminology is placed in articles. And it's nice to know this terminology. We do a lot of defining of words here. So it's good to understand the terminology that even the mainstream media is tossing around. You know, before the term RO, okay, or replication Z as it's known, the rate of reproduction of a virus, the rate of spread, we introduced before the media really started using that. So that's one example. But it's important to understand when they start saying, well, this is a virus, COVID-19 is a virus that is genetically modified. Well, what does that mean? That it is RNA based. Well, what does that mean? That there's a genome sequence. Okay. You know, to most of us, it's kind of like, look, I'm not a scientist, I'm not a researcher, you don't need to throw all those things around. Just tell me, do you have a vaccine and does it work and when is it going to be here? That's what's in it for us. Well, in the news that came out a few hours before the live stream, at least in the discussion coming out of pretty much both Canada and Israel, their timelines that they're quoting is several months, 4 months, possibly November, following human trials. And we're going to lay out what are really the 4 phases of those trials. You know, what does a drug have to go through? And it's going to be very simple, just the step, just in a graph. So we'll define what is DNA and RNA very simply. We'll go into talking about what's a triphosphate, what are the building blocks, what are the nucleotides, just by labeling them, just by citing them. We're not going to go deep into the chemistry and the biogenetics and bioengineering that's involved. It isn't necessary. But I'm pretty sure, I haven't started on it yet, but I'm pretty sure that the April edition of Entangled magazine is going to be devoted to what we're going to introduce tonight. We've spent a lot of time in past issues talking about third strand of DNA, dormant nanoparticles of DNA. We've talked about DNA replication, DNA modification, sequencing of DNA. We've really laid the foundation already in many past issues of Entangled. But I want to pull it all together, sort of my coalescence word that I use on occasion. In April, focused on the research that's going on, ongoing research, into vaccines and therapeutics. So this will be a foundation for the much deeper information that I typically provide in printed form in Entangled Magazine. And again, those of you that are not patrons are listening to this later on, if you join us as a patron, you're subscribed to Entangled Magazine. It comes out every month. Okay, let's get right to scripture for the evening. And Kathleen, by the way, my wife, won't be joining us this evening. She might come into the chat room a little bit later if she hasn't already. I know she's working on some other things related to her book. And as you saw at the beginning, let me bring that back up so that I don't just gloss over it. There it is. We introduced her book yesterday, the voting for her book at Taleflick. Taleflick.com. She has received really a prestigious invitation to have her book in the running for an opportunity to have her book made into a motion picture. And if you go to Taleflick.com, you can vote. And if you're listening to this on Friday, Friday will be the end of this voting period. It was only a 2-day event, or 2.5-day event, I guess it was. What I'm showing on the screen is not the present countdown timer. That's from yesterday. But the point is, if you go there and vote for her book, Pigtails and Potters Field, by clicking on the blue button on the right-hand side, we would very much appreciate that. This will result in, if she is the winner, it will result in an invitation from Hollywood producers, even independent producers, to option her book and develop a screenplay. So she's put 10 years of her life into producing this book. It has received all, on Amazon, all 5-star reviews, and just glowing reviews. So it's a book that she's put a lot of work into, and I've read it 3 times, and I think it's an extraordinary bestseller. When you read it, and I know that many of the patrons have already read her book, it reads like a bestseller. And she's a first-time author. She's self-published. So anyway, I hope you'll help us out with that. And I know many of the patrons yesterday indicated to us that they had already gone to Taleflick.com and voted for her. So thank you for that. And again, we'll go to our scripture, which is Psalm 91. And I just took a little excerpt from there, nor the pestilence that stalks in the darkness. But I'd like to take the time to read Psalm 91. You are my refuge and my fortress. He who dwells in the shelter of the Most High will abide in the shadow of the Almighty. I will say to the Lord, you are my refuge and my fortress, my God in whom I trust. Surely he will deliver you from the snare of the fowler and from the deadly plague. He will cover you with his feathers. Under his wings you will find refuge. His faithfulness is a shield and rampart. You will not fear the terror of the night, nor the arrow that flies by day, nor the pestilence that stalks in the darkness, nor the calamity that destroys at noon. Though 1,000 may fall at your side and 10,000 at your right hand, no harm will come near you. You will only see it with your eyes and witness the punishment of the wicked. Because you have made the Lord your dwelling, my refuge, the Most High, no evil will befall you, no plague will approach your tent, for he will command his angels concerning you to guard you in all your ways. They will lift you up in their hands so that you will not strike your foot against a stone. You will tread on the lion and cobra. You will trample the young lion and serpent. Because he loves me, I will deliver him. Because he knows my name, I will protect him. When he calls out to me, I will answer him. I will be with him in trouble. I will deliver him and honor him. With long life I will satisfy him and show him my salvation. As we did last night with our scriptures, we as the children of God, we have tremendous power and protection. We should not be dismayed by COVID-19 or anything else in this world. We are being built up. We are being strengthened to a much greater degree than Satan is doing with his evil in the world. And as the scriptures indicated to us last night, it may seem in the short term that evil is profiting and is experiencing abundance and wealth, but it's short-lived. We know that. We know that this is setting the stage for God's judgment. Okay? Am I saying that COVID-19 is God's judgment? No, I don't know the mind of God. I don't know his plans. I only know that he will protect his children. Okay? I'd like to start with just the basics. Nope, we don't need that again. We had our fun with that. Come on. My program's a little glitchy tonight. RNA and DNA. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So if you look here left and right, we can see the obvious comparison. When we look at RNA, it's just the single strand. It's the half of the double helix. What I'd like to point out to you is the sugar, the double-stranded sugar and phosphate. We're going to be talking about phosphate tonight. Okay, when we get into discussing RDRP, we're talking about RNA-dependent RNA polymerase, and that is focused upon phosphate. Phosphate is a salt. Okay, what does it say in Scripture about us? We're the salt of the earth. We literally are. We're the phosphate of the earth, if you like. Now, if you look around this graphic, you'll see that the basic building blocks of DNA and RNA. We see the typical cytosine, guanine, adenine, but uracil is one you may not have heard of, and then there's the thymine. So we're going to see where uracil fits into all of this. So that's just a quick snapshot. Again, I'm going to be pretty introductory in where we go with all of this. Just to backstep a little bit, again, this is the sequence. So bear in mind this graphic. This is the RNA genome sequence of COVID-19. We've shared this several times. And the 4 HIV inserts of short strands of the amino acids, the nucleotides, the building blocks of HIV. Okay? When we talk about DNA and RNA, this is getting down into more of the technical details, but this is just foundational to what I'm going to show you in a progressive graphic showing you how things work together between DNA and RNA specific to RDRP. So again, we have the phosphate here that's indicated. This large molecule here is ribonucleic acid, RNA. This is a sugar. And then the uracil that I just mentioned that replaces thymine. Thymine is in the DNA, uracil is in the RNA. Now you'll notice something here that I read to you last night, but I want to highlight, and that's this 5 with an apostrophe next to it. Let me see if I can bring this up a little bit larger. That's an indicator, that apostrophe after the number 5, and you'll see it after the number 3, is what's known as 5' or 3'. That shows the beginning and the ending of the sequence of the nucleotides that make up either RNA or DNA. So they are the beginning and end points to a string of those nucleotides. All right? Now, I'm giving you the components because we're going to get to a process where we're going to see where it is specific to a possible vaccine for COVID-19. That's where we're going with this. The 5 and the 3 mean 5' and 3', which indicate the carbon numbers in the DNA's sugar backbone. The 5' carbon has a phosphate group attached to it, and the 3' carbon is a hydroxyl group, okay, an oxyhydroxyl group. And I want to mention something before I go too deep into that. One of the interesting things that has come out is that there is a compound called hydroxychloroquine. Hydroxychloroquine. Kind of broke it up into 3 words there for you. Hydroxy, chloro, and quine. That's Q-U-I-N-E. Hydroxychloroquine is in the running as a possible vaccine and a therapeutic. I'm not going to go into that tonight, but I just wanted to have you make note of that because there's some late breaking news in the research into hydroxychloroquine. And I will bring that to your attention probably on Sunday in the live stream, and certainly it'll be included in the April edition of Entangled, where I will present probably 3 or 4 of the primary and leading possibilities for a vaccine. And I stress possibilities because there's so many moving parts to all of this. And there really are so many unknowns with COVID-19, but they have a distinct edge in developing this vaccine. One, the research coming out of Israel that I'll focus on in a little bit is already years into development. It was development for the purpose of Ebola, a vaccine for Ebola. And it appears that there's an opportunity to divert that research because it's so closely associated chemically, biologically, to COVID-19 that they may be able to fast track the existing research that they have and piggyback on that and develop a vaccine that originally was supposed to be intended for Ebola. And I'll get to an article on that in a little bit. So going back to this 5' and 3', the ends of RNA and DNA, the asymmetry gives a DNA strand a direction. The DNA polymerase, it works in a 5' to 3' direction. And this will make more sense when I put up another graphic here in just a second, and I'll get that brought up so that it makes sense. Again, the program I use here is not the fastest with bringing things up. Come on. Oh boy. Okay. This is where we're going. Remdesivir, RDRP, which is an RNA-dependent RNA polymerase, and ATP, which is adenosine, which is one of the nucleotides, adenosine triphosphate, meaning that it has 3 atoms of phosphate. Okay? Come on. There we go. So these are some in the phosphate group. We have a phosphate group and we have a nitrogen base in the adenine, and we have the phosphate group that binds with the sugar. So we're looking at the chemical layout for the bases that are made up of nitrogen. And then over here we have the phosphate group that is binding with the sugars. And it's really the ATP that we're going to focus on tonight, but you can see the others of the nucleotides that make up the phosphate group. Up here is what you would typically see in a genome sequence. So this is familiar to all of you, but this is where those letters are derived from. They're derived from the nucleotides that make up the genome sequence. Okay? Again, just kind of doing the building blocks here. When we look at the nucleotides, or they're also known as nucleobases, we have a nucleic acid that are polymeric macromolecules assembled from nucleotides, the monomer units of nucleic acids. The purine bases adenine and guanine and pyrimidine are base cytosine and occur in both DNA and RNA, while the pyrimidine bases thymine in DNA and uracil in RNA occur in just one. So I'm breaking those out for you so that you can see their chemical compound structure of these nucleobases. Again, just so you're familiar with where some of these terms, these nucleotide names are coming from. Okay. It may be that I have so many graphics that this program's kind of slow tonight. Come on. Here it comes. This is where we're headed. So this is where those nucleotides come into play. This is where we get into a genome sequence. What I'm going to offer to you is to show you, and I'll get to the punchline right here, is where RDV-TP, okay, the TP is the triphosphates, and the RDV, okay, is what we really want to focus on. RDV is the remetisphere. The remetisphere, RDV, it binds with the RNA molecule as it's being copied. When it does that, when it binds with this nucleotide, it stops what is known as the elongation of RNA. So moving from the left, from 3', the beginning of this viral RNA, which is COVID-19, and moving to the end point, which is the 5' end of the RNA, RDV-TP actually attaches itself. And then 3 or 4 more steps here of nucleotides being added on to the right, moving to the right, the elongation of the RNA. The RDV-TP actually stops the process of elongation or growth of the RNA. It stops the growth of the RNA of COVID-19. So that's where we're headed with this. I wanted to just give you the punchline, and then we'll go ahead and we'll backfill, as I often do here. Okay? So I've got all of this written out. It's a little bit complex, so I've got some notes that I'm going to read to you. The RDRP, or RNA-dependent RNA polymerase, this creates RNA from the 5' end to the 3' end of RNA. What it does is it reads the viral RNA template from the 3' end to the 5' end. Again, it's growing from 3 to 5. from left to right. This is the template of RNA that then produces our RD, that's RNA dependent, RP, or RNA polymerase at the top. And they're opposite. You'll notice 5' begins here and ends at 3' on the right. So they're a mirror image. They're a 180 of each other, if you want to look at it that way. So here's the process. We have a viral RNA as the starting point, the template. So these are already in existence. All of these amino acids, these nucleotides, these are already in place. What's happening here is RDRP is reading the U. It lines up its A, lines up its C, lines up its G. And when it is RDRP is sensing a U, it places an A at the beginning point of the growth at the 5' end. When it senses a G nucleotide, it places a C into its string, into its genome sequence. When it senses a C, it places a G there. Now, up here we have what are known as floating nucleotides, and they each have 3 phosphates attached to them. Okay, this is your guanine, your erythrosine, and your adenosine. And they each have 3 phosphates, but these are free floating in the bloodstream. These will attach, these triphosphates, they will attach. So you will have, for example, G in our sequence here, as it continues in the template, should have a C above it. Okay? I don't have a C up here to show you, but if there were a triphosphate nucleotide with a C, it would place itself right over the G. It would follow the normal sequence. The C would have a G over it. The G would have a C over it. If there were a U, it would have an A. But in this case, the A, which is the ATP, which is adenosine triphosphate, is actually overridden. It's overridden by the remdesivir triphosphate, the RDVTP. So this gives you an example of how the research takes place when they're talking about working with genome sequences. Well, what are they actually doing? Well, it's a process of substitution. It's a process of affinity. In chemistry, you would call it valence, the affinity of electrons to either give up an electron or attract an electron. But here we're talking about the actual molecule. We're actually talking about a triphosphate that is a nucleotide that has a stronger attraction, has a stronger affinity, a stronger binding. So it interrupts this adenosine, ATP, which you see up here. Here's the adenosine triphosphate that is a free-floating nucleotide. Ordinarily, it would float over here and place itself right over this U. But because RDVTP has a stronger affinity, stronger attraction, it moves in ahead of ATP. And then you'll see, if you extended this out further, if you went 3 or 4 more building blocks to the right, to the 5' direction, it actually stops that progression of the development of the RNA. It interrupts the sequence of COVID-19. I find this quite remarkable. I mean, this seems like it's such a simple explanation as to how do you actually deal with a virus. Well, within 1 week of the genome sequence being published on the internet by China of COVID-19, laboratories had already downloaded and started working on interrupting the genome sequence of this viral RNA. And this is exactly what we're looking at right here. This is what they're doing in the laboratories. Now, I'm going to share with you, or I did share with you before the live stream, an article coming out of Canada in which they are using plants to develop a vaccine. And that's a whole different approach. So we'll get to that later. So just to review here, RDRP sees the A on the first strand at the bottom here, and it creates a U. It then sees the U and puts in the A. It sees a G and puts in the C. It sees the C and puts in the G. When it gets to U, it should be an A, which is the ATP. But remember, remdesivir binds to U better than ATP will. Remdesivir binds as this RNA molecule is being copied, and about 3 or 4 nucleotides down along the line, it will arrest this elongation. It causes the RNA-dependent RNA polymerase to stop. Okay? The adenosine triphosphate is very similar to RDVTP, the remdesivir. Their structure is very similar, and I think I have a graphic for that. I want to show you, and I shared this with you last night, this graphic. Let's see if we can get this up here. Okay. Interesting. Give me just a moment. I'm going to post another graphic from my picture files here. Well, I apologize. I guess it didn't download. All right, let's move along. We'll get that back up there. I know this is a little bit cumbersome here, but I think we can make our way through it. So what we're talking about here is that RDVTP binds and fits well into the spot where ATP should be going. And what it does is it actually, if we look at the cytoplasm or the cytosol of a cell, we have the cell membrane and then the cytoplasm, as it's also called, and then the nucleus. You can take a look at that there. But the concentration of it is absorbed into the cytosol. And that's the place where this RNA replication, this elongation process, is actually stopped. It's interrupted within the cell. So when we talk about the perforation of the cell, and we've discussed and looked at how 5G does that, and it perforates the cell membrane and inserts the genome sequence of COVID-19 into the cytosol. This is where RDVTP actually enters as well into the cytosol and interrupts that activity, that replication of the RNA of COVID-19 within the cell itself. So again, it's kind of like, okay, well, where do we attack this virus? Where is the research focused in attacking it? Because viruses are not alive. They're not living, and they're not dead. They're just free-floating organisms. And I shouldn't even call it an organism because it's not living. It's just a material that is trying to bind itself with living material. And that binding process occurs by penetrating the cell membrane and then attaching itself to the cytosol of the living cell. And then it replicates using the energy from the cytosol, from the cytoplasm. It takes the energy from the cell in order to replicate itself. And then it continues to spread from cell to cell to cell. Okay? Alrighty. A little bit of terminology I want to share with you before we get into a couple of articles. Something called gene expression, and this is where the gene's information is converted into the structures and functions of a cell. When we say genes information, that's the sequence. Those are the building blocks, the nucleotides, where they come together into a sequence that form into the structures and the functionality of a cell. Closely associated with that is the transient gene expression, which is a temporary expression of genes, a temporary sequence, if you will, a temporary production of a cell. These will come into play when we get into a couple of articles here just in a moment. I wanted to show you a picture under a scanning electron microscope of what COVID-19 actually looks like. And if you see the magenta colored, sort of the pink colored items here in this picture of a living cell, this is COVID-19 itself moving out of a living cell. That's also called the SARS-CoV-2. That's a severe active respiratory syndrome, severe acute respiratory syndrome, SARS. So COVID is a further adaptation or mutation, if you will, of SARS itself. So if you want to know what it looks like, there it is. Okay. I know this is pretty dry stuff. I'm not being too expressive tonight. Just trying to go through these details as simply and clearly as I can, because I mean this can get to be very convoluted, and I'm trying to resist getting too deep into that. Again, when we looked at the uracil, which was the U that RDVTP attaches itself to, uracil is this nucleobase right here. And that's where RDVTP attaching itself to uracil stops that genome sequence from elongating any further. Okay? It goes to the 5' end, beginning with the 3' end. Now, with that background, let me go to a couple of articles. I'm going to go ahead and put this back up there in terms of gene expression, and we'll move on over to the late breaking news of what's going on. Okay. me give you just a little bit more depth on RNA-dependent RNA polymerase, the RDRP. This is a replicase, is an enzyme that catalyzes the replication of RNA from an RNA template. Remember the bottom row there, from the template. This is in contrast to a typical DNA-dependent RNA polymerase, which catalyzes the transcription of RNA from a DNA template. Alrighty, I think we've got enough of the basics there. So it's an interruption of that elongation phase of the RNA of COVID-19. Now with remdesivir, I want to share an article that just came out today, just this afternoon. This is from the National Institutes of Health, NIH. Clinical trial remdesivir to treat COVID-19 begins. And let me correct my statement. I was reading the date incorrectly. This is from the 25th of February of this year. The study enrolling hospitalized adults with COVID-19 in Nebraska. A randomized controlled clinical trial to evaluate the safety and efficacy of the investigational antiviral remdesivir in hospitalized adults diagnosed with coronavirus disease has begun at the University of Nebraska. The trial regulatory sponsor is the National Institute of Allergy and Infectious Diseases, NIAID, part of the National Institutes of Health. Okay, so I'm going to skip down through this. I sent this to you, so you already have a copy to be able to look at it in detail if you choose to. Many of you have heard of Dr. Anthony Fauci from the National Institutes of Health and also from the Centers for Disease Control, CDC. He states a randomized placebo-controlled trial is the gold standard for determining if an experimental treatment can benefit patients. So I'd like to put up another graphic for you on that. And here are the phases of clinical trials. Preclinical, then phase 1, 2, 3, and then FDA review, and then phase 4. Phase 1 is 20 to 80 participants, and that's where the drug is approved for testing in humans. Now prior to that, there's testing in, if you will, in test tubes, what is known as the when we're talking about outside the body, when we're talking about test tubes or petri dishes where it's testing before it goes to human testing, in the media, and especially in articles, you'll see them use the Latin phrase or the Latin term in vitro. In vitro just means outside the body. Now inside the body is in vivo. So when they go to drug approved for testing in humans, that's the in vivo stage of these clinical trials. Okay? Again, you'll run across this terminology, so I want it to be, somewhat familiar to you. So in vivo is inside the body. And then phase 2 is 100 to 300 participants. Phase 3 is 1,000 to 3,000 participants. And then the drug is submitted for FDA approval to confirm the safety and effectiveness of the drug. And then phase 4, the drug is approved, and that will be distributed to 1,000 or more participants in the continuation of the trial in phase 4. All right, so with that in mind, we'll just kind of go back to this article. A randomized placebo-controlled trial is the gold standard for determining if an experimental treatment can benefit patients. Clinical trials of remdesivir is also ongoing in China. NIAID developed, that's the National Institutes, developed the current study taking those designs into account. and in accordance with consultations convened by the WHO on the development of a therapeutic trial for patients with COVID-19. I'm skipping down through this. Just again, for those of you that are not patrons, if you would like a copy of this article, I'll send it to you if you contact me through anthonypatch.com. Initially, investigators will compare participant outcomes on day 15 in both the remdesivir group and the placebo group to see if the investigational drug increased clinical benefit compared to the placebo. All right, now again, I'm skipping through, just pulling some of the things that are most relevant to what I think you'd like to know. Okay. So that was in February 25th, where clinical trials were beginning with remdesivir for COVID-19. Well, let's jump over to Israel for a moment, and then we're going to go to Canada. And this is dated March 13th. Is that correct? Well, they're ahead of us because it's Times of Israel. Israel made oral vaccine for coronavirus on track, but testing will take months. Again, I sent this to the patrons. State-funded Miguel Galilee Institute has been working for 4 years. This is what I was referring to earlier. Has been working for 4 years on a vaccine that could be customized for various viruses. So it had a head start when COVID-19 emerged. This is very short. I'm just going to read you the body of it. An effective Israeli-developed vaccine for coronavirus is on track to be ready for testing within a few weeks, though it won't be available for months because of the lengthy and sometimes bureaucratic testing and approval process, a member of the development team said Tuesday. Chen Katz told the Times of Israel that the new oral vaccine for adults and children could turn this disease into a very mild cold. He said that for many people who are inoculated and then infected by COVID-19, potentially it will not affect them at all. So inoculated means they're vaccinated, okay? That allows the body's immune system an opportunity to build up a resistance to COVID-19. And then if they're exposed and they contract, they're infected with COVID-19, it will lessen the impact of it if they've been inoculated, okay? The rapid potential progress by the state-funded Miguel Galilee Research Institute stems from the fact that the institute has been working for 4 years toward a vaccine that could be customized for various viruses, and has now adapted that work to focus on the coronavirus, he said. Nonetheless, while Israel's science ministry made headlines last week by touting the institute's work and saying that its vaccine could be 3 months away, Dr. Asher Shalman, the health ministry's director of international relations, has warned against placing false hopes in it. The vaccine will consist of a specially produced protein, and Katz said he expects to be clutching a bottle of it within a few weeks. But then comes clinical testing, which will take place in conjunction with a partner, and the paperwork, both of which will take time. Katz, biotechnology group leader at the Institute, said, "By the time the protein is ready, we hope to have found the right partner who can take us through the clinical stage. The clinical testing experiments themselves are not so long, and we can complete them within 30 days, plus another 30 days for human trials. Most of the time is bureaucracy, regulation, and paperwork. Time could also be lost because of waiting points between the different stages of the process until regulators give the nod for things to move forward." So again, that's why I have the graphic up showing you the different phases. He spoke on the excitement that his team felt that it realized that the research they had been engaged in for 4 years could be tweaked to combat coronavirus. "The opportunity is amazing here," he said. "Everyone wants to know we can contribute something to humanity, and when we found we had the right tools to do it, this became very exciting." Katz's group at Israel's state-funded Miguel Institute has become a source of hope to many around the world, since it revealed on February 27th that it is working on the vaccine and said it hoped to achieve safety approval in 90 days. For 4 years, the research of Katz's team had been focused on developing a vaccine that could be customized to various viruses. It was piloting it with infectious bronchitis virus. But as coronavirus swept China, started adapting the vaccine for COVID-19. Its February 27th announcement prompted a widespread expectation among the public that people would soon be protected against coronavirus, which prompted Schulman's warning against false hopes. Katz clarified that the 90-day time frame in the February 27th statement was until the product is ready for testing, and he still believes this is realistic. He said that skeptics should understand that his team is not working on new research, but rather customizing an existing innovation, meaning that a fast turnaround is realistic. He stated the important thing is that we are working on a vaccine unrelated to this outbreak, and this is a great advantage. So I won't read the rest of this, but you get the gist of what they're saying. All right, I'd like to move on to Canada. This just came out today from Human Vaccines and Immunotherapeutics. I think this is the one the media is really going to jump on. Okay? Plant-derived virus-like particles as vaccines. So normally when a vaccine is developed, as many of you know, chicken eggs are used. Okay? And that's a very long, laborious process, especially when it comes time to scaling up of a vaccine using chicken eggs. It sounds kind of archaic in this, this day and age when we're talking about genome sequencing and gene expression and interruption of the RNA sequence and all of those kinds of things. It sounds kind of archaic to say, well, we're still using chicken eggs to produce a vaccine. Well, here they're going to a plant-based system. So I am going to take a little bit of time to read on this because it is, I think it's new for most of us to even be thinking that you could develop a vaccine from a plant. So I'll read the abstract. I'll just take excerpts out of it. I won't get into the real nitty-gritty of the language here because it just isn't necessary. Virus-like particles, or VLPs, are self-assembled structures derived from viral antigens that mimic the native architecture of viruses, but lack the viral genome. Okay? So they're taking the antigens out of the virus. We're not actually working on the genome this time, so we're stepping away from what we started with this evening. VLPs have emerged as a premier vaccine platform due to their advantages in safety, immunogenicity, and manufacturing. The particulate nature and high density presentation of viral structure proteins on their surface also render VLPs as attractive carriers for displaying foreign epitopes. Consequently, several VLP-based vaccines have been licensed for human use and achieved significant clinical and economical success. Let me skip down through some of this. I want to get right to the nitty-gritty. The advantages of plant expression systems. Okay, again, this is gene expression. This is converting the building blocks into the actual cells, okay, and the mechanisms of the cell. The advantages of plant expression systems are discussed in light of their distinctive post-translational modifications, their cost effectiveness, their production speed, and scalability. So again, we're moving away from eggs and we're going to a plant-based system, okay? Let me jump right on down through this. And I sent this to you for those of you in the scientific community or otherwise that would like to read the real nitty-gritty of this language that is in this peer-reviewed white paper. If you really want the background details, have at it, jump into the article. It's lengthy and it is very detailed, and I think it does a good job, however, of explaining why a plant-based vaccine direction may actually be the way to speed things up. If the Israelis are not successful with their direction in terms of working with the actual genome sequence, then this may be another opportunity in a completely different way to develop a vaccine. And again, let me underscore, I said it last night, I'm trying to take more of a positive direction in the discussion of vaccines. And the reason for that is I'm hoping and I'm praying that this vaccine that is produced is devoid of all of the things we've talked about over the years regarding the problems with vaccines. That hopefully, let me put it this way, that it's a pure vaccine. And that's why I'm spending so much time focusing on a vaccine, even though for a long time now I've been so down on vaccines. Now, does that mean that they're going to be honest with us about what's contained in the vaccine when they develop it? I don't know. I'm trying to go the benevolent direction with this. I'm trying, if you will, to put a positive spin on it. That doesn't mean that I'm not focused on watching out for indications that the vaccine may be more than advertised. And you know what I mean when I say that. So it's not like I'm cheering and saying, hey, everybody run out and take this vaccine. We still need to be very discerning. We still need to do our homework. We need to understand what is going on in the production of a vaccine. As much as we're able to glean, we obviously don't have the inside track on what's really being done. We only have these published white papers to go by and some of the occasional technical articles. But we'll do the best we can, use our discernment through the Holy Spirit to really try to come to terms with this vaccine that will be produced at some point. So I'm giving you the various methodologies in the research for a vaccine. And then we'll go from there, okay? Plants offer an attractive alternative system for VLP vaccine production owing to their ability to produce large quantities of recombinant protein at low cost. Their eukaryotic processing machinery for the post-translational modification and proper assembly of proteins and the low risk of introducing adventitious human pathogens. Plants do not require expensive fermentation facilities for biomass generation or the construction of duplicate facilities for scale-up production. Hence, plant biomass generation and upstream processing capacity can be operated and scaled up in a flexible, capital-efficient manner that cannot easily be matched by current fermentation-based technologies. When they say fermentation-based technologies, they're talking about using eggs. Okay? Several VLPs were initially expressed in plants and yielded encouraging results. However, these earlier attempts suffered from several drawbacks, including low VLP expression, plant-specific glycosylation of glycoproteins, and the lack of demonstration of producing VLPs with more than one protein. All right, I'm going to skip on past some of their problems that they had. The development of plant virus transient plant expression systems has overcome the challenges of VLP production speed and yield. As reported by our group and others, the cloning and high-level transient expression of plant-derived VLPs is simple and can be achieved quickly in 1 to 2 weeks of vector infiltration. Now this is going to sound a little strange, but vector infiltration with a tobacco mosaic virus, TMV, RNA replicon. This is a geminoviral DNA replicon system based on bean yellow dwarf virus. Importantly, these new improvements in the speed and yield of VLP production also provide the plant expression system a critical feature of high versatility for producing VLP vaccines against viruses that mutate their surface antigens rapidly. That's what COVID-19 is doing. It is mutating its surface antigens. And let me just stop for a second and give you a little bit of a news update. If you haven't heard already, there are now 2 strains of COVID-19. And this is going to be, a little deep, but bear with me. From the National Center for Biotechnology Information, which is the NCBI, the present genome label, the number system that they assign to COVID-19, its first strain, which is known as the reference genome, that is NCO 45512.2. Now that doesn't mean much to us, but there's a new strain and its number is MN99653.2.1, and it's known as the RAGTA13. The reference genome, the first strain of COVID-19, is ATG113. Now the second strain is RAGTA13. Okay? So just for those of you that like the technical stuff, that want specifics, those are the names of the now 2 known strains, predominant strains, the mutations of COVID-19. Now in other countries around the world, they're beginning to report more than 2 strains of COVID-19, because remember the natural process, even if this is a man-made construct, but it takes on a natural process, is to mutate based upon environmental influences. And so it's to be expected we will see multiple strains, multiple types of COVID-19. And so these guys have to take this into account when they're developing these vaccines. It's just like when we chase a different strain of flu every flu season. You get a flu shot, but it may not be specific to the strain of flu that suddenly materializes because of mutation. These things tend to mutate faster than the vaccines, than the quote flu shot can be produced and distributed. Okay? All right. Now, what I wanted to emphasize here is that they are using tobacco, what they call a tobacco mosaic virus, TMV, RNA replicon. When they're talking about plant-based, they're talking about using tobacco. Kind of ironic, I think. Conventional vaccines are produced by a costly downstream process and require continuous refrigeration, referred to as the cold chain, for their transport and storage. Similar to the storage and transport of fruits or dehydrated foods, fresh or dried plant parts containing subunit vaccines may present an ambient, temperature-stable product, thereby providing a possible solution to the transport, storage, and delivery of vaccines. At least in theory, oral immunization can be achieved by simply ingesting subunit vaccines in edible plant parts. This approach is appealing because it may eliminate or reduce the need for the costly downstream processing and allow plants to serve as a novel delivery vehicle for vaccines, in addition to providing a robust production system. Furthermore, the needle-free delivery method and natural preservation of vaccines in plant tissue may circumvent logistic challenges and allow practical implementation of immunization programs in regions where the cold chain and other medical supplies are limited. General concerns for the efficacy of orally delivered subunit vaccines include the possible denaturation and degradation of antigens by the digestion system, poor recognition of certain vaccines at mucosal immune effector sites, and whether edible vaccines would cause inappropriate antigenetic tolerance. Okay, so they go on and describe how this process using tobacco with VLPs and distributing and introducing it to the body through an oral absorption. Now, they don't say it here, but I would imagine that what they're going to indicate is if this is taken orally, they're going to want people to keep it in the mouth, probably up to 2 minutes. This, in other literature I've been reading on other approaches to an oral vaccine, the necessity is that you need to keep the material in the mouth for up to 2 minutes. Because the first level of introduction, let me get back to the camera, the first level of introduction of the virus is to the mucosa, to the lining of the nose, the nasal passages, the sinuses, and the oral areas of the mouth, and the first introductory areas to the the bronchial passages. So above the vocal cords, at the branching of the trachea and the esophagus, that area, and to the lips. And so probably they're going to have people either let it dissolve in the mouth or chew without swallowing to break it up and maximize the absorption into the mucosa, into the linings, and then go ahead and swallow it and allow it to be ingested into the stomach and then into the bloodstream. Part of what I think they're going to do orally is to have people keep it in the mouth, as I said, and then breathe in deeply. And that's going to be kind of tricky. You don't want to be choking on this. But the idea here is to allow yourself to breathe the medication into your airway passages, into your bronchial tree, and deep into the lungs. Because it's the lining, that mucosa, the lining of your bronchial airways, that first protect your lungs. And this is where COVID-19, and I've mentioned this before, COVID-19 first attacks the lining of the mouth and the nose and attacks the lining of your bronchial airways. It strips it away, and then it's able to attack the bloodstream, okay? So I think this is where they're going to go with this oral medication. Let me jump down through this a little bit here for you. Again, I appreciate this is pretty dry tonight. It's not our usual fast, rapid fire, delivery of a lot of things. It's kind of detailed and kind of dry. But hopefully this will just give you a snapshot as to where some of this is going. Okay. I want to get down to their summation. Enveloped VLPs are shells assembled from the capsid and or the envelope proteins of enveloped viruses surrounded with a lipid membrane known as the viral envelope. Now, some of the discussion regarding COVID-19 is penetrating a double wall into the cell, a double lipid membrane. And this is part of the problem with the vaccines, is the vaccine being able to penetrate, as does COVID-19, into the cell itself, into the cytoplasm, okay? Into the cytosol of the cell. The presence of a host cell-derived membrane provides additional possibilities to integrate heterogeneous antigens and adjuvants, either embedded in the membrane or enclosed inside the lumen, but also potential challenges in regulatory approval due to the uncertainty of host cell components in the envelope. So they are indicating that using a leaf-based system will allow them to penetrate the cell. Okay? Leaf and plant culture cell derived introduction into the cell. Again, I'm summarizing some of this. It's very deep. I'm just trying to give you the highlights that mean something to you. Okay? The plant-derived pandemic H5N1 VLP vaccine has been tested in a phase 1 and phase 2 human clinical trial, and an H1N1 VLP seasonal vaccine candidate has been tested in a phase 1 trial. So plant-based regarding those two viruses, H5N1 and H1N1, those are already in phase 1 and phase 2 trial. And kind of like the discussion with Israel's approach, where they were working on Ebola and a customized a treatment or a vaccine that could be customized to a specific type of coronavirus, which there are hundreds actually of coronaviruses, types of coronaviruses. The ability to have one that can be customized to a specific coronavirus is advantageous, and they're already up into the beginning of human trials. And with the plant-based, they're approaching phase 2 human clinical trial as well. So it looks like, in terms of research for a vaccine, there at least are a couple of approaches that already are well along in the trial process. Okay. Not sure that the rest of this is going to be germane to us tonight. Let me read this last part to you. Downstream process development should aim to increase manufacturing productivity, reduce production costs, enhance scalability, and preserve the integrity of the VLPs and ensure the compliance of the manufacturing procedures with the FDA's regulations. Human clinical trials of plant-produced VLP, successes in the production, purification, and demonstration of strong systemic and mucosal immunosensitivity have prompted several clinical studies examining the safety and immunogenicity of plant-produced VLP-based vaccines in humans. Okay, so what they're saying there is, when I was talking about the mouth, the mucosa, the mucosal immunization, the lining of the mouth, and in particular the nose and the sinus cavities. A phase 1 clinical trial has also been completed for a seasonal flu vaccine candidate based on plant-derived HAVLPs of an H1N1 strain, also known as the swine flu. So they're already into phase 2 human trials with plant-based VLP research. I guess that's the best way for me to summarize this. I don't want to get too involved with this because it really does get pretty dry. I don't mind reading it, but I don't think you want to hear all of it. You guys can read it if that's of deep interest to you. Go back to my notes here on some of the news stories. I mentioned this to you yesterday, and I want to kind of wrap up tonight going back to Gilead. This is from 27 February of 2020. Gilead launches remdesivir's phase 3 trials in COVID-19 patients. And I'll put that back up here if I can find it. So phase 3 is 1,000 to 3,000 participants. This is where the trials are with Gilead. Many people had heard about Gilead regarding its stock price shooting up about 2 weeks ago. So we'll kind of look at what they're doing here. Gilead Sciences has commenced 2 phase 3 clinical trials of its investigational antiviral drug, remdesivir, for the treatment of COVID-19 in adults. This announcement comes after the FDA accepted and reviewed the company's investigational new drug application for remdesivir in this indication. The randomized, open-label, multi-center studies will assess the safety and efficacy of the drug in nearly 1,000 patients. A 5-day and 10-day dosing regimen of an intravenous formulation of remdesivir will be tested. The trials will mainly be conducted at sites in Asian countries impacted by the coronavirus outbreak. From March, sites will be added in other countries with a higher number of confirmed cases. One of the trials will enroll around 400 patients with severe clinical manifestations of the novel coronavirus disease, while the second trial will involve about 600 patients with moderate clinical manifestations. In addition to standard of care, participants will be given 200 milligram dose of the drug on day 1 and followed by 100 milligram dose every day until day 5 or 10. Both trials will evaluate clinical improvement as the primary endpoint. The primary endpoint in the first trial will be measured by the normalization of fever and oxygen saturation. The second trial will record the measure based on the proportion of patients discharged by day 14. Gilead Sciences Chief Medical Officer Merdad Parsi said Gilead's primary focus is on rapidly determining the safety and efficacy of remdesivir as a potential treatment of COVID-19, and this complementary array of studies helps us give us a more expansive breadth of data globally on the drug's profile in a short amount of time. The initiation of phase 3 trials comes a day after the National Institutes of Health at University of Nebraska Medical Center announced the trial of remdesivir. The 2 studies led by the China-Japan Friendship Hospital are already being conducted in Hubei province, the epicenter of the outbreak in China. Okay, I think that was pretty much all of the news items I wanted to get together here, with the exception of Canada. I sent this to the patrons as well. This comes from Defense One. Headline, and this is from today, Thursday the 12th. We've got the vaccine, says Pentagon-funded company. Canadian firm says that it could make 10 million doses per month if its innovative production method wins FDA approval. A Canadian company says that it has produced a COVID-19 vaccine just 20 days after receiving the coronavirus's genetic sequence using a unique technology that they soon hope to submit for FDA approval. Medicago, that's M-E-D-I-C-A-G-O, Medicago, CEO Bruce Clark said his company could produce as many as 10 million doses a month. If regulatory hurdles can be cleared, he said in a Tuesday interview, the vaccine could start to become available in November. An Israeli research lab has also claimed to have created a vaccine. That's the one that I already outlined for you. But Clark says his company's technique, which has already been proven effective in producing vaccines for seasonal flu, is more reliable and easier to scale. There are a couple of others who are claiming that they have, well, we call them vaccines for COVID-19, he said, but they're different technologies. Some are RNA or DNA-based vaccines that have not yet proven in any indication yet, let alone this one. Hopefully they'll be successful. How did Clark's team create one so quickly? They used plants, not chicken eggs, as a bioreactor for growing vaccine proteins. Traditional vaccine production requires eggs. So we went through the technical white paper that is underlying this news report that has come out about the Canadian approach to this, okay? So Medicago doesn't work with live virus. Instead, it uses plants, a relatively new approach that has seen much advancement in the past decade. It inserts a genetic sequence into agrobacterium, a soil bacteria, which is taken up by plants, in this case, a close cousin to tobacco. The plant begins to produce the protein that can be used as a vaccine. If the virus begins to mutate, as is expected for COVID-19, they can just update the production using new plants. That's the difference between us and egg-based methods, he said. We go directly to producing the vaccine or the antibody without having to propagate the virus. So what they're doing, instead of growing the virus and then attenuating or lessening its strength, its virality, is to actually produce a protein from the plant that will then act as a vaccine, okay? Using plants and genetically engineered agrobacteria works faster than eggs and also makes the vaccine much easier to produce at scale, which in part is why the U.S. military has invested in the company. And this is where DARPA put together $100 million for the program, and that program is called Blue Angel. I'll skip down through this a little bit. Okay, our basic plan is to be in human studies phase 1 by the July timeframe. And then it would depend quite extensively on the decisions the regulators make in terms of the hurdles they want us to have in the normal course of development. Clark says he understands that cutting corners in drug development invites risk, but, he says, there's a lot of room for negotiation with the regulators. I won't put words in their mouths. I will say our intention, taking a very standard approach, is that by November, we will have completed phase 3 in clinical trials, allowing the vaccine to be made widely available to the public. Okay? So, that's considerably shorter than the 12 to 18 months that we had been hearing from Dr. Fauci from CDC. And I hope that gives us a glimmer of hope, if it is truly a purely benevolent vaccine. But again, our discernment. Okay, let me make sure I haven't skipped anything here. So we have two approaches, the plant-based and we have the remdesivir genetic process of interrupting the elongation of the viral RNA by inserting remdesivir TP, RDV-TP, into the sequence, binding with the U, if you will. So I hope I didn't completely lose you guys on this, and I'm not being condescending. Again, I always say that. It's just, it is very dry material. The sum total of what I'm presenting to you is that there is hope here for a vaccine. There is hope for a cure, and the other concurrent product is the therapeutic for those that are infected. I pray and I hope that this will be a pure process, that this won't be a mark of the beast process, this won't result in the medical tattoos that we have presented, that this will be mankind, rising to the challenge and doing this for the sake of humanity, doing it in a benevolent fashion and not in a population reduction scenario that we've often, brought up here. I don't know. The question often comes, is this it? Is this it? Is this, the pestilence? Is this the bowl judgments? Is this the end of days scenario. I'm not trying to belittle that. I'm not trying to water that down. But I think we all go to that direction of wanting to see the positive for humanity, not the destruction of humanity, that mankind can rise above the greed and the selfishness and the power struggles, rise to the occasion and put an end to this. It certainly has not only decimated lives, it's killed people, it's destroyed people's lives just by contracting it. And certainly economically, it's destroying the world. And we've spent a lot of time, including last month's, well, I should say the March issue of Entangled magazine, talking about the economic impact here in the United States predominantly was the focus of the magazine. But I just pray that we can rise to this. We can get over ourselves, if you will, and get back on track economically, get back on track with our health, back on track with hopefully improving the environment. So I've tried to put more of a positive spin yesterday and today on this whole COVID-19. We certainly raised the alarm bells, and we talked about, without fear, talked about being prudently prepared to deal with the economic situation, specifically the disruption in the supply chain economics based on outsourcing production of an awful lot of what we consume in the United States in China. And a lot of other countries are dependent upon China. Now, if we can trust any of the numbers coming out of China, we are starting to see numbers being reported indicating a decline in cases in China. Now, that may be falsehood, that may be, the Chinese Communist Party, the CCP, trying to put their positive spin on it, and certainly domestically trying to put a positive spin so that they can get people to comply. I mean, they were forcing people, but now to get people to comply with going back to work, not just sheltering in place, not just, quarantine, self-quarantine and social distancing, but actually people thinking that it's on the decline and so it's okay to go back to work in China. So we can't totally trust the numbers. I certainly don't trust them. I find it very hard to believe that with the millions of people in China, I mean, there's well over 1 billion. I think it's 1.4 billion or something like that in China before COVID-19 hit. Hard to imagine with that number of people that really they would be seeing such an early decline in the numbers. So I'm not believing it. And I guess that kind of flies in the face of putting a positive spin in terms of talking about a vaccine, but I think it's too early. Now let's get to our domestic, and that is the president talking last night about his measures, his steps, and I think you're all familiar with those. One of which that I'd like to highlight is the essentially closing of the border to Europe, not allowing flights to take place, airline travel of flights coming out of Europe, except for the UK, which boggles the mind. Excuse me. But the idea is that he's trying to close the borders, trying to minimize the numbers of people coming into the country from Europe. And that includes Japan. That was done first with China, and now it's with Japan and Indonesia and Europe in general. And I applaud that. I think that's a good move. But I think there's another step to that. And I know this is going to sound conspiratorial. This is going to sound sensational and alarmist. But now that COVID-19 is so widespread, there really isn't, short of having a vaccine, there really isn't anything we can do except to try to minimize the movement of people and the close contact of people. Again, the social distancing, 6 feet apart from other people, sort of thing. In the spirit of that, of trying to limit the movement of people, I think the next step that the president needs to take is to seal off the southern border, especially, okay, the border with Mexico, and perhaps even with Canada. I don't think it would be difficult with Canada because we have good relations with Canada, and we have a strong border in Canada. And we don't have the numbers of people trying to cross the Canadian border into the United States that we see in the southern border. About 3 days ago in California, in Southern California, at the border crossing, American intelligence reported that there were large numbers of people on the Mexico side, the Mexican side of the border, that were gathering to stage a rush on the border into California. And there were about 150 U.S. Army troops deployed to support the Border Patrol. Active duty U.S. Army troops deployed along the southern border in California. I think for a short period of time, I think, again, it is prudent on the part of the president to deploy the U.S. military. Yes, National Guard, but also active duty military across the entire southern border of the United States. Because you can certainly see how circumventing the restriction on air travel coming out of Europe, that circumvention can be accomplished by flying into Mexico and then crossing into the United States. So I would anticipate that will be, if you will, phase 2. And I think that's a reasonable thing to do. It sounds like quarantining. It sounds like they're trying to cut us off. It sounds like they're trying to trap us within the United States. There's lots of ways of taking that and looking at it that way. And I'm well aware of that. But when we talk about trying to control the spread, it's by controlling the movement of people. And I think it just makes logical sense to close the borders. I don't think people should be fearful. I don't think they should panic over that kind of a thing. I think it would be a short duration move on the part of the president. Now, it's easy for people to go and, blow that out of proportion and run off with scenarios. And I'm not going to do that. It isn't necessary. I'm just going to say I think the president needs to close the borders, at least for 30 days. Then perhaps we can internally, domestically, get a grip on this. Because what's coming next are the test kits. And we've talked about that. And there are 2 approaches to testing. There's a nasal swab or an oral swab. Okay, that's one. It's a swabbing process, typically deep in the back of the mouth, with a long Q-tip. And you've probably had this when you've gone in to get a physical, or if you've had, the symptoms of strep throat, and a long Q-tip is inserted, and it's sterile. It's inserted deep into the back of the mouth, into the throat, and a swab is taken, and then that's inserted into a tube, sealed, and sent to the lab to culture that in a petri dish. That's one way of doing it, and the other is through actually extracting blood samples. In either case, however the sample is obtained, then a genome sequencing approach is taken to compare, as I indicated to you, the sample, the original sample of COVID-19, and now the second strain that's been identified and labeled here in the United States of COVID-19, to use those as reference sequences of the genome. And that's where the testing really takes place, is examining the sequence. And this is part of the reason I wanted to share these kind of dry discussions about, the nucleotides and the sequencing and the arresting of the elongation of the viral RNA by RDVTP. Because this is what's going to come out in the discussions in the media about testing. So you know more when the media just simply says, well, it's genetic testing or genomic testing. They're testing the genes, and that's about as far as the media will go. Now you understand more of the background as to what that testing really involves when they're looking at the gene sequence. And why are they looking at the gene sequence? The answer to that is so they can interrupt that process of replication of the RNA. Okay, so that's your takeaway for tonight, is if it's remdesivir or if it's a plant-based virus that can be taken, vaccine that can be taken orally, it really is that interruption of the, not just mutation of the virus, but more specifically, more early, before mutation, interrupting the growth through the expression of the sequence. It's kind of like Occam's razor, the simplest solution. When I was sharing with you the interruption of the elongation of the RNA, that's what occurred to me. It was like, boy, this is a very simple solution. However the vaccine comes out, whichever one, or there may be a couple of approaches, we could see concurrent distribution of 2 or maybe 3 different vaccines. Widespread, not beyond phase 4. Okay? I don't know. I don't know how they're going to approach this. It could be that we end up with large portions of the population in those hot zones. Hot zones defined by clusters, number of cases. Those hot zones being the obvious target for the human testing beyond laboratory trials, beyond phase 4, going into the actual general population to test the vaccines. Because remember, they're fast-tracking this. And again, I'm trying to be positive about this. I'm trying to look at it in a benevolent way. Fast-tracking to get it into the general population means that they're going to go to those clusters of already affected cases, populations, and test the vaccine in those clusters before going to the more sparsely numbered cases in the general population itself. That's what I would expect to see coming. Testing in clusters. That's a simple way, short way of saying what I'm expecting to see happen. I don't think that's bad. I'm just saying that I expect that to be the technique for rolling this out in a fast-track manner with 1, 2, or 3 probable vaccines. Because in order to truly fast-track it, you have to present it to large numbers of people. Yes, that renders us as guinea pigs. If we happen to be in a place like New Rochelle, New York, with a 1-mile diameter circle where they have identified a cluster and they've closed it off, or as one of our patrons in the chat room tonight lives in the Seattle area, that certainly is a hot zone. That's a very large cluster. And certainly I would expect that the first vaccines will be distributed into the Seattle area because it has the largest numbers right now in the United States. Here in the Boston area, we're seeing a cluster as well, not to that degree, but the governor of Massachusetts has declared Massachusetts in a state of emergency, and certainly Boston is in a state of emergency. That doesn't mean people are running around like crazy here out on the streets. It's pretty quiet. It's pretty quiet out right now. It just means the declaration of an emergency allows resources to be targeted to the problem, okay? Federal resources in particular. All right? So I hope this gives you more of an upbeat view of what's going on. I'm not saying these things just to try to placate people, just to try to present a false hope. I'm trying to be factual in my reporting for you. And I'm sure on Sunday I'll have a lot more to report on COVID-19. I think it is going to be the primary topic for obvious reasons. Associated with reporting on that will be the economic impacts that are going on. So I will be monitoring that. Tamara has been sending me large numbers of tweets regarding Syria and Iraq. One thing to point out is right now U.S. troops are going after Iranian-backed militia, not in Iran, but in Iraq itself. Right now, as we're speaking, there's a battle within Iraq against Iranian-backed and equipped militia in Iran. So, in Iraq. So Iran is still at the forefront and is still a target, still a problem. And Syria is still a hotbed. None of that has calmed down. And in fact, it's escalating in Syria. And it's moving quickly towards, it appears, moving quickly towards the Golan Heights. Okay? So we're not totally distracted away from the Middle East by looking at COVID-19 and the economic issues. But we'll pay attention to that, and I'll post articles that are relevant and important as we approach Sunday. And Lord willing, we'll be back together on Sunday. So always a lot to report. Again, I apologize. This is kind of a dry presentation tonight. I think it's just the nature of the material that I was going through. Your big takeaways are that there are at least 3 significant approaches to developing a vaccine. It's genetically targeting. It's fast-tracked to looking anywhere from 4 months to November for human trials to begin. Okay? Some are already with associated research into human trials, phase 2 and phase 3, that can be adapted, research that can be adapted specifically for COVID-19. Again, fast-tracking things. I guess in summation, it looks like maybe November, maybe we might see a vaccine. Let's hope it doesn't have a lot of garbage in it. Let's hope it isn't all that negative stuff we've talked about before. And if this behaves like a more natural virus, maybe with the warming of temperatures here in the United States and elsewhere in the northern hemisphere, this thing will follow the course of typical flus, and it will go dormant for the summer months. It's been reported, and I think this is valid, that we may be stuck with this as a chronic situation every flu season, that it's just going to be another flu, so to speak, that we're going to have to deal with. But I think we can be pretty prepared for it if it does come back around. I think developing quickly a vaccine, one or more types of vaccines, will give us the ability to be inoculated and to thereby be able to diminish the effects should we be exposed in the next flu season that comes around. And hopefully during the summer months approaching the next flu season, economically, the world can get back on its feet. Okay? So, I pray and I hope this is not the mark of the beast scenario. I pray that this is not the third strand of DNA scenario I've talked about since 2013. I'm not moving away from any of that. I've already said all the negatives out there about this whole situation. I'm just trying to give you a different approach to it, a more positive approach. Okay? I thank you for all of the information you guys have been sending to me. We did not talk about colloidal silver and structured silver tonight because I wanted to focus just on the vaccine research. Feel free to continue sending me information about silver. We probably will talk about it on Sunday not in a real technical aspect, but briefly, I think Sunday we'll touch on silver a little bit. Okay. I want to take the remaining 10 minutes for questions in the chat room. I've not been able to pay attention to it, and I know Kathleen's really busy with other things, so I don't think she's been in the chat room either. That's why I've kind of just went on and on and on, because I didn't have the opportunity to take comments from you, during this presentation. Let's see. Sapphire reactor. Very good. There is a new video from the Sapphire project. I haven't watched it yet. It's been out about 2 or 3 days on YouTube. Sapphire project has an update, and I really want to make time to watch that, because I really, really, really applaud their efforts with developing a new way of producing energy that is modeling the sun, and the primary objective was to model the sun in a realistic way, in an accurate way, beyond the theoretical, where science has been trying to tell us that the energy of the sun, I'm going to be very short with this, but the energy of the sun comes from consuming hydrogen within the sun itself, when in fact Sapphire has demonstrated empirically, experimentally, that it's drawing from the plasma of the universe. It's drawing energy from the cosmos into the sun, and that's what's continuing the processes within the sun. But I'll maybe do a live stream on Sapphire after I've watched their update to see what their latest developments are. But Monty Childs I've communicated with. It's been some time since he and I've corresponded, but Monty Childs and I, early on in the Sapphire project, we exchanged an awful lot of ideas, and I had a great time putting my 2 cents in to try to give them, some ideas. That's all I was doing, just offering ideas of what they might try and what they might see. And I'll tell you one thing real quickly. They did find the tetrahedral pattern within Sapphire. They did find the 600 cell tetrahedron within Sapphire. Pretty remarkable. Okay, so I'm glad you guys brought that up in chat. I'd love to spend a whole live stream talking about Sapphire. I think it is probably one of the greatest experiments that's been done. Okay, so Debbie is saying that Trump has been exposed 2 times already. So 2 times to COVID-19, I guess is what you're saying. And I will make a personal observation. All of us were watching the television. We don't own a television, but my in-laws do, and we were watching President's address last night after last night's live stream, and we all said he looks very tired, if not ill. And we conjectured that possibly he does have COVID-19. And I did read something today very briefly that apparently he's refused to be tested for COVID-19. I mean, that's the story I read. I don't know how true that is. But it wouldn't surprise me. I mean, with all of the traveling he does, all of the public events he's been involved in, all of the meetings with groups of people that he has, at the White House and other places, how is he going to avoid it? How's he going to avoid contracting it? I mean, the guy is a public figure. He's out in the public all the time. So I pray that he does not have COVID-19. I want to emphasize that. Okay. Debbie's saying that President Trump was with the president of Brazil, who was positive with the virus. Okay, well, that's not good news. Sorry, I'm coughing because I've been talking for 2 hours straight. Let me see what else you guys have going on. I'm going back up through the chat room just to see what you guys have been talking about. If you have questions that I don't address here right now, go ahead and send them to me by email, anthonypatch.com in the contact tab, or go to anthonypatchauthor, AUTHOR, @gmail.com. And I respond to pretty much every email that I get. Okay. Okay, guys. Thank you, Deborah. I appreciate your comment in the chat room. Alrighty. I guess we'll go ahead and wrap this up. I think there's hope. I mean, all my notes here that I wrote up today, my takeaway for myself is I think there's great hope for a vaccine. The question for myself and for my wife and our family is, are we going to take the vaccine? No. Kathleen's in the other room. She said, no, we're not taking the vaccine.
I don't have hope for any vaccine.
I don't know if you can hear her, but she said she doesn't have any hope for any vaccine. Okay. Anyway, I don't want to take a vaccine. I pray that we will all remain healthy and not contract it through the power of the Holy Spirit, through the power of our prayers. And as I said with our scripture this evening, He covers us with His wings. Okay, so we don't have any fear over it, and I don't think that we are going to be in that group of population that is going to need a vaccine, because we won't have the signs and symptoms of COVID-19, so we won't need to take it. Is there going to be a mandate? Will it be mandatory for everyone? Well, that's the scenario we've talked about at length before, that it'll be mandatory. Possibly it will. If it becomes truly widespread into the hundreds of thousands of people in this country contracting it, and large number of people dying into the thousands, I think we can pretty much expect that they're going to mandate everyone taking the vaccine, and everyone having that tattoo that we've talked about, the medical tattoo, with your medical records and your vaccination records applied to the skin, under the surface of the skin. Let's just pray that it doesn't come to that. I guess that's really my mindset. Let's just pray that it doesn't come to a mandate to take a vaccine. I would like to opt out of it. Kathleen would like to opt out of it. Okay? So, be prudently prepared. Take your reasonable precautions. I would say, yes, follow the suggestions by the CDC. And limit your contact with people, and proper hand washing. Avoid touching the face, the eyes, the nose, and the mouth. Okay? Take your vitamins, take your vitamin C, take your vitamin D. There's a number of articles that came out today about high dose vitamin C may be effective. I don't know how effective it's going to be. But vitamin C and vitamin D certainly is not going to hurt you. There's an old saying that when you take a lot of vitamins, you have very expensive urine. Vitamin C, typically in most metabolisms of most people, the body can only absorb about 500 milligrams of vitamin C in 24 hours, and the rest is secreted from the body. Okay? But taking 500 or 600 milligrams of vitamin C every 24 hours is certainly not going to hurt you. Okay? One last thing about vaccines relates to the flu vaccines. There's a lot of discussion going around in the media about people should take a flu vaccine. Well, which strain of flu are we talking about, number one? Therefore, what vaccine are you talking about? But as you all are well aware, when you take a flu shot, no matter what strain it is, it's a attenuated, a weakened form of that strain of flu. And so it produces a low-grade fever, sometimes a cough, sometimes a productive cough and a sneeze, and generalized weakness or malaise and generalized weakness and achiness in the joints and muscles, because you actually have contracted the flu, but to a weakened level by taking a flu vaccine, a flu shot. Well, I've seen media stories out there in the mainstream saying it would be a good idea to take a flu shot so you don't weaken yourself by contracting the flu, whatever strain of flu, in which you then weaken your body, weaken your immune system because you contracted flu, which then opens you up to COVID-19. So take a flu shot is what the media is saying, so that you don't develop those flu-like symptoms that open the door to COVID-19. Well, that's counterintuitive. I think it's counterproductive. I don't think you should take any flu shot. The reason being, it causes an immune response. And the killing action, the killing mechanism of COVID-19 is a cytokine, not a cytosine, which is a nucleotide. It's not cytosine, it's a cytokine with a K, K-I-N-E. Cytokine, which is an inflammation and an inflammatory response by the immune system, and it's called a cytokine storm because there's so much inflammation, fluids produced in the body. It's actually retaining of water, builds up in the lungs. That's the pneumonia. It's the cytokine storm that produces the pneumonia. Well, if you have a low-grade flu because you were injected with a flu vaccine, that produces a cytokine, an inflammatory response by the immune system. That's normal to try to flush that flu strain out of your body. Produces white blood cells, encapsulates the flu strain, and flushes it out. Okay? I don't think that's a smart thing to take a flu shot thinking that it's going to somehow prevent you contracting full-blown, full-strength flu, which then would open you up to COVID-19. I think any level of immune response by the body at this point, due to a flu shot or the flu itself, whatever strain of flu, if you already have a cytokine response taking place in your body, that opens the door to COVID-19. So I think the best approach is don't take any vaccines, eat well, take your vitamin supplements, follow the hand washing and all of those other recommendations through CDC, the social distancing, and limit your travel. Just reasonable and prudent steps. I think that will go the longest way towards preventing you being exposed to someone who has COVID-19. Now, I know that there are reports in the last few days that COVID-19 may last for an extended period of time just in the air, that it's airborne. Well, I think more studies need to be done on that. More testing needs to be done to show the veracity of that statement or that claim. But I think certainly being in close proximity with people, especially ones that have signs and symptoms of a cold or flu, if not COVID-19 itself, is the best defense. Just staying the heck away from other people. Okay? And if you're around people, the hand washing, don't touch your face, don't rub your eyes, take your supplements, try to stay in fresh air, Try to be in a well-ventilated area. Okay? Alrighty. So we've been 2 hours. I want to go ahead and be respectful of your time and not drag this out any further. Okay? Again, send your prayer requests. We honor every one of them, and we are honored to do that for you. And we thank you for your prayers for us. And again, please, if you would, go to Taleflick and vote for Kathleen's book. It ends sometime on Friday. I'm not sure exactly what time, but spread it around on social media. No one's going to contract COVID virus by voting for a book. Okay? And as I said last night, if you find yourself needing to self-quarantine, or as I prefer to call it, shelter in place, maybe you'd like a good book to read. Okay? Pigtails and Potters Field. It's a great story. Thank you, and God bless you all. We'll talk to you on Sunday. Bye-bye.